Hello, this is Chapter 17 of Applied Statistical Analysis for Psychology and Human Factors, and today we're going to be covering multi-way ANOVAs, or multiple predictor ANOVAs, in contrast to one-way ANOVAs. That's going to be Chapter 17 of the course book. You can get the course book at this website under course textbook right here. The link is in the description. And Chapter 17 starts around page 395 or so. So previously we've looked at regression, putting categorical variables into regression, and now we're going to look at just extending this further, and putting multiple categorical variables into regression. You can follow along if you go down here, follow this link, download this Chapter 17 walkthrough, which I've done, and I have it located here. Make sure you set your working directory to the source file location. So we're going to start with looking at the tooth growth data that we've looked at several times before. And again, this is a data set that looks like a 2x3 design. It's got different doses and it's got different supplements, and we're going to treat dose rather than as a continuous variable. We're going to treat it as a categorical variable. And one thing to think about, sometimes it's hard to realize when you see data like this that the data aren't in a 2x3 table. Anytime you're doing an ANOVA or regression, you're looking for data in this format where you have a dependent measure and multiple independent measures. And the independent measures are coded as different variables, and what it's going to do is make combinations of the levels of these corresponding to these different points. But you don't need to make the table yourself, because then you lose all the information about the variability within each point. So anytime we do an ANOVA, we're really still looking for data that are sort of in this long format. So I'm going to recreate this dosage variable that we've done in the last chapter. And we'll just do a simple regression and look at the summary. So previously we just thought about the one-way ANOVA, where you put a single categorical predictor in, and maybe partly for computational reasons, partly for historical reasons, this was often thought of as its own test. And you still see a one-way ANOVA as its own separate test in many like GUI software packages where you look at menus. And last time we also talked about a number of non-parametric tests, and many of those will only handle one-way ANOVAs. So it does get a little more complicated when you have multiple predictors, but just as you're comfortable with regression having multiple predictors, it shouldn't be that much more complicated to add multiple predictors in an ANOVA. But let's just do it as a regression here, and just like we've done before, we're going to use plus. So length tilde supplement plus dosage will allow us to have multiple predictor variables. And if I do a summary, I can see here, this, remember, shows this shows me the main intercept, and then instead of each one of these is going to be treatment coded. So it doesn't have a supplement OJ. That's considered the value, baseline of value of 12.45, and then this codes off of orange juice. And then you have dosage 1 and dosage 2. It doesn't have a dosage of 0.5. And so the intercept is really the orange juice with a dosage of 0.5. And you can see, well, that really is 12.45. Although that doesn't actually look right. If I were to if I were to add a zero-intercept to this, maybe we can look at this. Well, 12.45 is a supplement for juice, and it would be for zero dosage. Maybe this it looks like it's a little higher, but maybe that's just a artifact of how it's plotted or how I'm looking at it. So if you add, here's the interesting, if you add this zero-intercept, it forces the first thing you say to sort of take the role of the intercept. So now each supplement has its own intercept, but dosage still is relative to that level. So if you do enough of these, you get to learn how this is coded. Again, so if this is zero, we have two intercepts, and then dosage will be relative to this. So dosage 1, the model says it's 9.13 higher than either of these. Dosage 2 is 15.5 higher than either of these. Let me get rid of this 0 and go back to here. And again, now it's a little more complicated, but 12.45 is the baseline. We subtract 3.7 to get the baseline for vitamin C. So that's this difference. And then we add 9.3 to get these two, and we add 15.49 to get these two. Just to look at what that might look like, we could create the model, the fit to the model, and plot that fit. And this is really what the model is suggesting is its best estimate. And you can see that, you know, it doesn't account for this, what looks like might be an interaction where they get closer together, they converge as the dosage gets larger. It has no ability to do that because we just incorporated additive effects of each. But you can see that the difference here is not linear. It permits having a smaller difference here than here. These are, after all, independent levels of a factor, and so it picks its own mean. So it picks, so this picks sort of the best difference between the two, and you can see it underestimates these two because it has to overestimate this one. But otherwise, it seems like a pretty reasonable model, I guess. So that's what happens if you look at the linear model. But oftentimes, if you want to test it with an ANOVA, you're really asking, overall, is vitamin C different than orange juice? Or really, are vitamin C and orange juice the same, or do they lead to different outcomes? And similarly, overall, does changing the dosage lead to the same outcome or different outcomes? So that, instead of just looking at the coefficient, we can test with an ANOVA, because then we're really doing a hypothesis test of whether that factor matters. So I can put this linear model into this ANOVA function, and it will give me, it'll answer this question. And now you can see I no longer have multiple rows for supplement, or I only had one row for supplement, but multiple rows for dosage. It doesn't tell me the coefficients anymore, but it does tell me whether each of these matters. You can see that the degree of freedom here is 1 because there's 2 levels here. The degree of freedom here is 2 because there's 3 levels here. It gives sum of squares and mean squared and the F-score. And this F-score is really asking whether supplement matters after everything else is taken into account, and indeed it does. Does dosage matter after everything else is taken into account? And indeed it does. So, but when you look at this, it's sometimes puzzling where all this comes from, and we're going to spend a little time digging into that, figuring out where each of these pieces come from, and maybe what some of the alternatives might be that might actually be reasonable things to do. So what if we had just done dosage? We had ignored the supplement type and we just done dosage. So what changes? So we look through here, well, you know, if we look, I guess we'd have to look up here at the coefficients. We've got 9.13 and 15.49, and down here we have 9.13 and 15.49. So these estimates don't change when we remove this one, and this is because we have an orthogonal design. There was, and so that's really nice. The intercept does change, but that's kind of fine because that's just sort of a baseline level anyway. And but in order to do this with an ANOVA model, we'd like to put it in the ANOVA. We could have done ANOVA on this like did before, or we could put it all in the AOV model. And we can do this for the larger model and the smaller model. And now there isn't a lot of, well, this dosage relation still has the same sum of squares, you can see. But the residual changes. And the residual changes because, well, here we've taken some of the sum of squares from the residual and put it in a supplement. So when you're adding more predictors to it, you're moving things from the residuals to things you account for. And that will have the effect of making this one, you know, more reliable, more significant. So we can, I think if we do a summary of each of these, this will give us the tests and more details. And you can see that here the F-score changes between dosage here, but the mean square error is the same. The p-value changes, although it's really strong in both cases. Sum of squares is the same. So some things are the same and some things differ. What you can't, and this doesn't show things like residual standard error, which you would have to get from the linear model. And so, let's see. So it still is sometimes reasonable to do the linear model of these to compare them. Here are the linear, and if you think about it, if we think about how dosage and supplement work together, and how we are comparing them with the ANOVA, remember how the linear model is just a single fitting, but then to test it, we test it with this ANOVA process where we compare it to another model. And it all depends on what the comparison model is. If you look at this model that has both, we really have several different things we could compare it to. We can compare it to the intercept-only model, and that's this one. We could compare it to the supplement-only model or the dosage-only model. Furthermore, if we want to know if supplement mattered, we can compare supplement to the intercept-only, or we could compare supplement and dosage to dosage only. Both of those differ by only supplement. And so we're gonna, these now sort of form a lattice of models. We've talked about this before in regression. They form a lattice of models that are sort of logically related by just adding one value to it or another value to it. So I could do that, or I could do the actual ANOVA versions like this. And this is the model you usually do, or this is the model you usually do. And so if we were to look at M both, summary of M both, or ANOVA of M both, or so summary of M both or ANOVA of M both, you see that it gives us this basically an F-test for each individual variable, But really this is a comparison, an F-test that's a comparison between models. So we can verify that what this is really doing is comparing this model to get the supplement, and this model to this model to get the effect of dose. By doing those ANOVAs by hand, let's see, and here is the corresponding comparison between dosage and supplement, and you can see that that this f-test is the same as the f-test that appears here. So if you want to know the effect of dosage, you're really comparing the model with dosage and supplement to the model with supplement. And we can do it again here. And so now if you want to look at this 14, it appears right here. So really what this ANOVA model is doing is it's taking neighboring models and comparing them to one another. And it sort of makes sense that you want to compare the model with supplement and dosage together with the one with just dosage to find out if supplement works. Now you could also look at the model with supplement and compare it to nothing. That's also asking if a supplement works, but it's not asking whether supplement is effective when you consider the effect of dosage. And that's important because sometimes, especially if these effects are correlated, you've got you could have both of them accounting for the same variability. And so you want to be, if you want to know if supplement works, you want to know that it works even when you account for everything else. So that's generally the strategy, although it doesn't have to be the only strategy you use, that to know if something works, you try to see if it works after everything else is accounted for. So this comparison will look at if dosage works, and this comparison will look at if supplement works. Alright, so that's the basic logic of how we do a multi-way ANOVA, and you can see it is a little more complicated because unlike a one-way ANOVA where you're just comparing that effect to the intercept only model, it's not clear which pairs of models you should compare. And as we'll see later, as you add more and more variables, there are more and more possible comparisons of models you could do. That might be interesting. Okay, but first, we talked about, for one-way models, how to do post-hoc testing to see if specific variables differed. And we can use the same libraries to do that with ANOVA models, with multi-factor ANOVA models. So if I take AOV with both, and I put it into the Tukey HSD, it reports the Tukey multiple comparisons of means, so family-wise confidence levels for both of them. So here are the differences in dosage, and here are the differences in supplement. So you can read these. Here there's only one of them, and you can see that this difference is significant according to the post-hoc Tukey HSD test. And each of these pairings is significant as well, according to that test at almost any level you would look at. The Agricole library also has this, and this one allows you, I think, to select, well, it allows you to make a table, and it allows you to, I think you can select which variables you want. So here I specify that I want the post-hoc test for dosage, and it gives me dosage. The means of these, and here is the whether they are the same or not. And because A, B, and C are each different, it sort of bins them and puts them in different bins. This one is really handy if you have a lot of variables and you want to say, and if you have 7 or 8 variables, if you just have this, it's sometimes hard to figure out which ones are different, which ones aren't, and so having this can help organize. Here's a set of 5 that are the same, and then I have one that's higher than everything else, and one that's lower than everything else. So that's the Agricole, and the Tukey test, HSD, and the built-in Tukey test. So this is just one set of post-hoc tests. There are a number of them that you might want to explore in specific circumstances, but in general, this is adequate for most uses. Okay, we also had the ANOVA Bayes factor ANOVA, and previously we looked at this with regression, and now we can look at it with categorical variables. And if you remember, some of these models in Bayes factor are really sensitive to having categorical versus continuous predictors here. They don't know how to mix them very well. But the ANOVA BF will handle multiple predictors here. And we can look at the summary. And this will give us, this nicely gives us the whole lattice of models. Well, I guess it doesn't give us the intercept-only model, but each of these are compared against the intercept-only model. It even gives us the interaction model, which we didn't ask for, but might be of interest. We'll talk about interactions, I think, in the next chapter, and so we won't think about that right now. But now, to test whether any of these matter, there's several ways of doing this. This tests whether supplement differs from intercept only, and this is whether dosage differs from intercept only, and this is whether supplement plus dosage differ from intercept only. But really, if we want the equivalent ANOVA comparison to the ANOVA, we want to see if this differs from this or this differs from this. So you can pull those out just like this. So this is the third level, and I can compare AB3 to AB2, and it'll even label this right. Supplement versus dosage has a Bayes factor of 56, so there's really strong evidence of supplement having an effect over and above dosage alone. And here is supplement dosage versus supplement alone, and again, really strong evidence of supplement plus dosage having an effect over supplement alone. So both of these are have strong evidence for being predictive according to the Bayes model. We use the same logic, but you have to sort of do some of it by hand. So you compare the more complex model to the one with everything else, because you want to know if it has an effect over and above the effect of everything else that you're using. All right. There's an exercise that I won't do right now, but we'll probably cover in class, or you can do on your own to get a little more practice doing this. And so this is all, it all seems pretty straightforward when you have an orthogonal design. And really that's what the analysis of variance was designed for. You know, I think a lot of it was designed by agricultural researchers who can clearly make all the levels of a pesticide, just like some of these examples we have. They've got really, they can really easily control these things, and they want to counterbalance sort of where the tree in the field is, so that they don't, so they can account for that, and so the designs can be really complete and clear. But oftentimes we don't have this ability. So let's look at a sort of hypothetical data set, and we'll do a version that's orthogonal, and then we'll do a version that's not orthogonal. So here's what it would look like if we had an orthogonal design for, you know, this would be like a pretty standard psychological experiment where I vary color, and I vary set size, and I vary stimulus size. So maybe this is sort of a visual search task where I'm looking for something of a specific color in the context of multiple things of different sizes. And we want to see if any of these have an effect. And just in this case, I'm creating random values to see what they're like. So we wouldn't really expect to see anything that's significant anyway. But if I look at a table of my design, I can see that I've got, you know, one element in each group. So this should be, this design matrix is going to be, well, it's going to be uncorrelated. It's not going to be orthogonal unless I centered all of these. But if I treat them as categorical predictors, then it might also be orthogonal if those are coded in terms of orthogonal contrasts. So, but I guess right for here we're going to treat set size and stem size, in this case, as numeric predictors. And so you actually, yeah, and so sometimes if you have a numeric predictor that you want to treat as a factor, you know, you'll forget about this, and then you have to look at these coefficients and see, oh, there's only one set size coefficient. So although I have 3 levels, it's only fitting one thing. That's the indicator to you that you have a sort of a regression version instead of an ANOVA version. But for color, because I have 3 different levels, I've really got these 3 things. And so intercept codes the baseline for red. Oh no, for blue, and then we have green and then red. It's in the opposite order as that I put it here because it's going to be in alphabetical order. And so this, if I do a summary of the linear model, it gives me all the coefficients, and it gives me this residual standard error and the f-test. Remember, this f-test compares everything versus the intercept only, which is probably not what I care about. I mean, some people report this because it sort of tells you if there's anything there or if the whole thing is better, but you shouldn't just by default report this because it's usually meaningless. You know, it's only meaningless if all these individual pieces were significant, and so why not just report each of these? Color, set size, and stimulus size. But if I do the ANOVA, it collapses each of my groupings into a single line, to a single row. And so really it's really going to collapse color into a single row. And it also ignores intercept, right? So you don't really need to consider intercept here because the intercept is not about comparing levels. I don't know why I'm pointing here, but it's about everything's going to be compared to that intercept only model anyway. So this gives me the table, and remember, now each of these is going to be the effect of this in the context of everything else. So I can test whether color has an effect, and set size has an effect, and stem size has an effect. And I could have easily made these factors and treated that, and then there would have been 2 degrees of freedom in each of these as well. Okay, so let's say we wanted to test the effect of set size here using an ANOVA, and remember, just like we said before, set size is we want to test the difference in set size by computing an ANOVA between adjacent models, one that contains set size and the one that doesn't. But within this context, there's a lot of ways we could do that. And it's sort of a tough question because you know, set size isn't significant and color isn't significant here. So do I want to test the set of, so do I want to test this one, just looking at set size alone? Well, that's not significant. Or do I want to test it, so this is testing it with respect to the intercept. Or do I want to test it in the context of color? But color wasn't significant. Now you can see the F value here differs. Although, let's see, the change in mean square error, or the change in some square error here, is going to appear multiple times. So we still see this 0.127 in each case, but the other things change because I'm accounting for more variability by adding them to the model instead of leaving them in the residuals. But it sort of doesn't, so other than, you know, by I could pick one of these and the sum of squares that I'm accounting for by stem size is going to be the same. I'm accounting for the same amount because it's, but that's only true because it's an orthogonal design. but you should also recognize that the f-test is changing each time. It's a little different each time. And that's because the residual error is being moved around. I'm moving it from the residuals that I'm comparing it to into things I'm accounting for already. And so if they account, now these never really accounted for anything, but if they had, that would take variability I know about and account for it and make my other effects more easy to detect. What happens though if, that's what happens if we have an orthogonal design. But let's look at a similar thing where we have a non-orthogonal design. And so instead of doing it in a design where, like I did above, I'm just going to randomly sample, and we'll pick a new set of response times. And now if I do a table of this, you can see that because I'm randomly sampling these, there's some with zeros and some with twos and some with threes and a lot with ones. And so it's, first of all, it's not even complete. I've got pairs of these that I don't have observations in. Now that's, in this case, kind of okay because I don't have, I'm not going to be looking at the interactions. But it's certainly not sort of a balanced design because of this, even though it has the same number in each group, the same number of observations in total. So I can do the same thing now, look at these same models, and now if we look at the delta sum of squares for each of these versions, set size versus intercept, set size and color versus colors, or set size and stem size versus stem size, and set size and color versus set size, color, and stem size. We see a different change in the residual standard errors. So these numbers aren't going to be the same in these cases. And that, you know, if you remember back to when we talked about orthogonal factors or orthogonal predictors, it's sort of the same issue. So this is an unbalanced design, and it sort of leads to a problem here, that the estimates we make, not the estimates, but the tests we have, sort of depend on the versions of the model we compare to one another. And so it's not just like when it's balanced like it was up here. We could get different statistical tests, but that's because we're moving variance around. Now in this case, we're even getting different changes in the variance that we're accounting for. So it's kind of a question, which one of these is the one you want, especially when some of these might not be a significant predictor. Do you want to include them or do you not want to include them? Now, we never looked at this before, but in the linear model, if I use these as predictors, I get the same coefficients. So they're in a different order. If I use the same predictors in a different order, you can see that this maps onto this maps onto this, and this maps onto this. And that's just because, you know, the optimization process, we're inverting that hat matrix, doesn't care the order. It doesn't have anything to do with the order. So a linear regression model will estimate the same coefficients no matter what order you put them in the model. And this is, some people will, it's a little confusing because sometimes there's notions when you are doing hierarchical testing of the order you enter the variables in. So it doesn't matter for the coefficients you're estimating, but it does matter in the ANOVA test because the order they're in here depends, dictates in R how those comparisons are made. And because we just established that those differences in sum square error are going to change depending on how you put it in, you'll see differences. So when you use the lowercase ANOVA function or the AOV function, which is the same, to look at whether particular effects have a result, you will get different results depending on the order you put the predictors in. The coefficients remain the same, but the ANOVAs are different. This seems a little puzzling. I mean, this puts it in a different order. And in this case, they're not too different, but if you had high multicollinearity, you might see them differ by a lot. So this is maybe a little bit troubling, and there really is, this is just a fundamental aspect of doing the ANOVA model when you have orthogonal or non-orthogonal predictors. We could have done it even in the AOV model, and looking at the summary of the AOV model. It's the same as we saw before, and the two ways of making the model are different. So why is this happening? Let's break down all the different submodels again. So I'm going to come up with these 5 models: the intercept-only model, the color, the set size, and the color plus set size, and the set size plus color in the opposite order. These are just the raw linear models, and now I can do comparisons of these two. So if I want to see the intercept only versus color, I get this. If I want to do set size plus color versus set size, here are 2 different ways of testing the effect of color. And now you can see that they lead to different differences in sums of squares, different f values, and different p values. And so, because it's not orthogonal, the way we do this changes not just this, but this as well. Previously, with the orthogonal design, this didn't have an impact. This didn't matter. I mean, the order didn't. The way I did this didn't matter. We could do the same thing with checking on the effect of color, or on set size. You can see again we've got 2 different values here, 2 different f values and 2 different p values. So now if I just do an ANOVA on the 2 top models, let's see where these numbers come from. So I've got 0.17 here, and we can see the 0.17 here. 2.9, I'm not sure if the 2.9 are going to appear, but then 2.87 should occur. Well, if we do the right comparison model, we should be able to see it. The 2.24 appears here. And so the 0.24 appears when I'm comparing color plus set size to color alone. And the 0.17 appears when I'm comparing set size to the intercept alone. So really what's happening when I do this one is this is sort of being added to the intercept alone model, and we're taking that comparison. And then this is comparing color to everything else. And probably really what we want is this value for color and this value for set size. And so what this is called, like this is the traditional ANOVA method, but people have developed what they call a type 2 ANOVA. They call it type 2 ANOVA, and so they call this a type 1 ANOVA. So the type 1 ANOVA will take each variable one by one and remove it, and test them in sequence, and remove them in the order that you specify them in the model. And this is nice because it means all of your residual error gets removed one piece at a time. Your total residual error adds up to 1. But it doesn't allow you to do this logic of I want to compare each one of these set size in the context of everything else. And so that's the logic that I think it was generated in the SAS community. That's what it sounds like, and replicated in SPSS, and they call this either a type 2 or type 3 ANOVA, where the specific order won't matter because it'll always give you the effective set size after everything has being accounted for. So what we sort of want to do is pick out the bottom row of these two instead of the top row of these two, or any single one. So we want to know, for color, to do this, it's going to take color and then set size. So we want the effective set size from here and the effective color from here, because that sort of asks, what's the effective color once set size has been accounted for, and what's the effective set size once color has been accounted for. Okay, so there's some discussion in the book about how to add all these up and to prove that what's going on is proper. So, but what you can show is that for the standard type 1 ANOVA, it doesn't matter which order you do it in, you get the same amount of total sum of squares. You can add them all up, and in each direction they don't matter. But if we do this other way, we're going to get more sum of squares that we're accounting for than exist in the whole data set. So that's kind of weird, but it's sort of the compromise we make. You can use the drop command, or maybe the drop one command, to do this. So if you think about what the drop command did, it tried to compare we use this in linear regression, it tried to compare this model to every model one smaller. And that's really what you want to do. To know the effect of RT, or of color, you want to compare it to the model of set size. And to know the effect of set size, you want to compare it to the effect of color, so color plus set size to color. So the drop one command actually sort of implicitly creates this table, or the drop, is it drop one? Yeah, the drop one command creates this table. This is, I guess, a slightly different way of looking at it. But now we can see that, maybe if we look up here at the, well, it has residual sum of squares, the sum of squares, 0.24 and 5.82. I get the 5.82 from here and the 0.24 from here. So this is exactly what we said we wanted. We wanted this guy and this guy together. And so oftentimes if people are asking on the R helplines, how do I do a type 2 ANOVA, they'll say, well, just use drop one, which doesn't make a lot of sense. It seems like it's for a different purpose, but this creates the type 2 or type 3 ANOVA. And it shows the effect of each one. Now, but this one, I guess this won't give you the F and T test, but it gives you an AIC test, and you can probably get, maybe if drop one, if you do test is equal to F, then I can get the F tests out of this again. So this is sort of probably what you really want to do. Unfortunately, it's not the default, because sort of the ANOVA is just the traditional ANOVA, and so this creates sort of a problem that people don't recognize this and often use the wrong ANOVA function, or use the ANOVA function incorrectly with R, and because it always does this type 1 ANOVA. Now that's not going to matter if you've got an orthogonal design, but it will matter if you have a non-orthogonal design. So the standard solution in R is we'll create a library that does it right, and that's what a number of people have done. And one is the Easy ANOVA package has ways of doing this, but there's a library called CAR. It's companion to Applied Regression in R book, and it has an ANOVA with a capital A. And so if I do this linear model, these linear models, note that these are in a different order. With the capital A ANOVA, I get exactly the same answers between both of them. It doesn't matter, and it tells me I have a type 2 test. The ANOVA the capital A ANOVA will let me specify, it won't let me specify a type 1. I should, but you could do a type 3, which in this case I don't think is going to be, I guess it tells me the intercept term, but in this case it's not going to give me anything different. So what I would recommend is to load the car package and use capital A ANOVA whenever you want to do a real ANOVA, especially if you have a non-orthogonal design. It gives you sort of what the community expects and what you probably are expecting as well. Alright, so the final example is just going to build on this notion a little more to get you a little more comfortable with it. And we're going to think about the model lattice when we have a slightly more complicated one. In the textbook, I have a number of, let's see, at the end of the textbook, I need to, let's see. There we go. So in the textbook, I have a picture of this lattice, this model lattice that we talked about before. And so sort of the sample problem, let's say we have A, B, and C predictors. We're trying to predict Y. And we've looked at we thought about pieces of this, but this sort of shows how these are all related in this lattice. And this whole structure is a lattice. And every one of these rows represents a model of a different level of complexity. So the bottom row is the intercept-only model. And this first row is one with a single predictor, A, B, and C. Then the next one is A and B, or A and C, and A, or B and C, 2 predictors. And the last one is 3 predictors. And so if you are doing a standard ANOVA, you're sort of following one of these paths. And I think I have a diagram of this in the next chapter, although this is talking about interactions as well. If you're doing a standard type 1 ANOVA, you're following one of these paths. You're saying, okay, I have the complete model, then I want to test A, B. If I do A, B, and C, I'm going to test A, B, and C versus A and B, and then I'm going to test A and B versus A, and then I'm going to test A versus the intercept. So one of these directions is the type 1 ANOVA. The type 2 ANOVA, or type 3 ANOVA in this case, where they're when we don't have an interaction, is sort of these 3 comparisons. It's saying I want to compare this whole thing to each model one below it. Okay, so let's return to this more simple lattice. But what you can see is the transitions between any of these models is always a single predictor. And I've written on this graph the amount of variance, the sum squared variance that's accounted for these predictors. And you can see that because, well, in the data I'll show you in a second, they're not orthogonal, the amount that's accounted for differs in different contexts. And especially, like if you look at A, sometimes, like here, it accounts for 307 units, sum squared units, and sometimes it accounts for a lot less, 25 units or 31 units. Sometimes it's 300. And so it depends, the amount it accounts for depends on the context of what else has already been accounted for. It depends on whether B has been factored in. Similarly, B, the amount of variance B is much larger, and so maybe that makes sense that B is accounting for a lot of variance, that if it's already been accounted for, there's not as much left for A if they're slightly correlated, and the design is slightly orthogonal. And again, whenever you are testing the effect of A, you have to think about, well, if I've got all of these predictors, I have 1, 2, 3, 4 possible effects of A that I might be interested in. What's the right one? Generally, it's going to be like these three, but sometimes it might be these three, or it might be something like this, or something like this, and you just have to, and it just makes, it depends more on the logic and the design of the experiment and your knowledge of how the world works than anything that you can tell just from doing statistics. So the way I built this lattice is by running a bunch of models like this, and we can, let's see, let me create the variables here, and if you recreate this, you should be able to get exactly the same numbers. And so here's just the standard ANOVA, and you can look at the sum squared error is 31, 27, 414. And so you can see that ABC follows the C first with the 400. Well, if you start from the top, it's really starting from the bottom, I guess, but it's following 31, and then 27, 27, and 415. So that's this model. That's this ANOVA model. Another ANOVA model is a different pathway through this. So here is when I give ACB, this one's the same, but then I go to this path and it ends up being 592, I guess, and then I go to this path, 2551. So really what these are all comparing is, this is A versus the intercept only. This is C versus A plus the intercept only. This is B versus A and C and the intercept. So you can run each of these models and compare it and try to identify the pathway in this lattice that it's really following to help you get an understanding of why we might want to consider these three. Because these three are the ones that are looking at the effect after all of the other effects have been accounted for. All right, and then you could also, instead of doing it this way, you can compare specific pairings. So if I want A versus the intercept, I can look at here, and you can see it's -31. A versus the intercept is right here. So we can verify sort of where you are in the model lattice by doing these types of things. And this is a little different approach than is usually taught in a class like this, where you're just shown the ANOVA table and taught how to interpret it. But really, when you're doing an ANOVA, you should always be thinking that you're really just comparing nested models to one another. And there's a bunch of different ways you could do that. And sometimes this particular model you want to do is a subset of these, and it might be a different subset of these. So it might be these, but it might not be. It might be some other subset. And so that's sort of the logic of testing that you ultimately need to do. All right, so this is it for this chapter. If you look ahead at the next chapter, we're going to be talking about main effects and interactions and how they are tested and implemented within a factorial ANOVA.